Anyway, here we are. So today we're going to finish up this chapter and hopefully talk about when we can expect our first exam. Maybe a week from today. Yeah, question.
Are you recording this?
I am. So yep. So I'll post that recording as well too, which will probably be handy because this will be a
All the recordings
different.
up there, right? So like one class.
Yes, yeah. There might be like two that I didn't. There one or two that weren't on Zoom, but yeah, I'll try and record everything and put them all up there. So, or just embed my lovely voice over the slides, like I've done that before because that saves some bandwidth. Yeah, we'll see what I can do. Okay. So question?
Yeah.
Mhm.
Okay. So we talking about the metabolic pathway.
Yes. So this is chapter 15.
What is AMPK?
Yes. So our AMP, of course, is our adenosine monophosphate. Our AMPK is AMP kinase. So one of the most common and probably because it's also a very rapid way of altering one of the most common ways of regulation because it's so fast is phosphorylation or dephosphorylation of compounds, because you can get a rapid signal cascade that will just turn things on. And we kind of saw this at the end of last semester when we talked about epinephrine or adrenaline. We get this really quick, one molecule of epinephrine can result in 100,000 glucose molecules being released within a very short time frame. So again, our ATP levels, their flux is fairly significant, but if you lose, 10 millimolar of ATP, that might be, 16% or something like that. Whereas if you lose, I think actually the example is 5. Let's scroll back up here. Yeah. So if we have a 0.5 millimolar change of ATP, that's a 10% change. But for AMP, that's a 600% change. So we often see AMP being a more fine-tuned sensor of the energy state of the body. If we suddenly see this big increase in AMP levels, that's going to indicate a low energy state and start triggering processes to make more ATP. So for example, it will tell skeletal muscles in the heart to start bringing in fatty acids for energy because we're low on glucose. We can't do glycolysis. It's going to tell the liver to stop fatty acid synthesis and to start gluconeogenesis. It's also going to inhibit cholesterol synthesis and glycogen synthesis because those things will cost ATP as well too. And basically our AMP kinase is active in all these different organs, and depending on what those organs are doing, for example, if we're at low energy, we probably don't have a lot of glucose circulating in there. So it's going to tell the pancreas to don't secrete insulin because we don't have glucose.
So is that connected to diabetes?
Yes. So I believe there is what was that example? I think about last week where we had the rare form of diabetes where, yeah. But normally diabetes is, either insulin sensitivity or not making insulin. So in this case, if something messes up in here and we have constantly high levels of insulin being secreted, then we're going to see some other weird effects in there that are probably not diabetic, but similar. Okay. Now, our AMP kinase is affecting or regulating a lot of different enzymes, but some enzymes limit the flux more than others. Okay. I'm gonna subject you to my awful, and by awful I mean wonderful taste in music. And it always makes me think of the Smiths, a band from the 80s, Morrissey's out there on it, where there's a song, "Some girls are bigger than others." Some enzymes have more flux than others. All right, so whatever mnemonic sticks in there. Enzymes that are really, really far from equilibrium are the ones that are regulated, but they're not all regulated the same way. Okay, and you can think of this like if, a terribly bitter, cynical view of the legal system, not all crimes are prosecuted the same way. Not all enzymes are regulated the same way. Some will control flux through an entire pathway. Others will just help regulate the steady state concentrations of different metabolites in response to changes in flux. So maybe we're starting to build up a lot of fumarate. We might see an enzyme that helps regulate the fumarate, which doesn't really affect a particular pathway directly, but indirectly helps respond to changes in flux. If we look at hexokinase and phosphofructokinase 1, those are good targets for regulation of glycolysis, okay, glycolytic flux in here. Increased hexokinase activity will allow us to activate more glucose as it comes into cells, so we can kind of get that glucose charged with phosphate and move it on wherever it needs to go. Increased PFK1 is going to specifically send more glucose to glycolysis to make energy. All right, so what we'll see if we have a high glucose level in the bloodstream, our muscles, and of course the brain will be using this. Okay. So hexokinase, though, affects the flux through glycolysis a lot more than phosphofructokinase 1. Okay. And that's just because we're going to see a lot more charging of this, so we can shunt more of that glucose in there. Our PFK1 is, a lower, I don't want to say it that way, doesn't have as great a control over the flux within glycolysis.
That's one thing I was looking at the chart. Is this just in the body, like conceptually, like just in the body, or is this in like a certain region?
So yes, and we're going to talk about that more here in a minute here. But normally we're probably talking about the liver in most cases, because when you consume glucose, everything's going to go in the liver. And I always, as we get more into metabolism, I'll go ahead and do it now. I always used to think growing up, oh, the brain is the most important organ in the body. And now I'm like, it's probably the liver. yeah. And brain's a close second, but the liver is like the regulatory center in here. So we'll talk a lot about the liver. And that's just because, as we bring in fuel and as we work to excrete stuff, everything kind of passes through the liver. Liver is Grand Central Station. All right. The other thing on this chart, though, that's important, and when we see hexokinase has a big effect on glycolytic flux, phosphofructokinase, smaller effect, but significant effect. And then phosphohexase isomerase has pretty much no effect. And if you recall, phosphohexose isomerase is one of those other 7, enzymes in the glycolytic pathway, but it's reversible. So it just goes either way. So if we get too much, and that's why it's not a target of regulation, because it'll just go either way depending on the concentrations of products and substrate for it. Okay. And we can see get to a maximal level pretty quickly for our enzyme here. Half a unit of phospho-PFK1 were there, half a unit, 1 to 2 units of hexokinase. Anyway, don't worry about this elasticity coefficient. This is another slide I should probably get rid of, but our elasticity coefficient is really looking at our responsiveness, because we're looking at our initial rate when we just add a little bit of substrate versus when we have it saturated. And ideally then our elasticity coefficient should be 0, because it's just pretty much constantly loaded. But we could actually take a look at this slope at a low concentration and figure out how rapidly our substrate is binding. And if we change something about it, if we regulate that enzyme, we may see a change in that. So that would be how we define our elasticity coefficient. Okay. More importantly, what I really want you to take away from this is our regulated enzymes are quite often, almost always, the ones that will have the same substrate and product, but a different enzyme to go from one way to the other way. So glucose to glucose 6-phosphate requires hexokinase. If you want to go back the other way, though, you have glucose 6-phosphatase to remove that. So because we have 2 different enzymes, those are the targets for regulation.
ACE was added on to like everything that's going the first way. So like,
yes.
If I just added that to everything.
So generically, ACE is indicates an enzyme. So glucose is the molecule, glucase would be something that acts on it. Our kinases are ones that specifically, the kine is adding phosphorus. So kinase, that's going to be adding phosphorus. Our phosphatase are going to be things that take phosphates away. when I was first learning biochem, it always sounded like it should be the other way around, chi for cut, but no, it's reversed.
That
in
would
there.
be better.
Yeah. Phosphatase for putting a phosphate on that. And you'll see mutases induce a mutation. What else do we have here? Dehydrogenase. We're going to take away some hydrogens in there. We may see dehydroxases every so often. Enolase, we're just kind of interconverting in there from our but that, which is isomerase, mutase, enolase. They're doing very, very similar things. It just kind of depends on where they were discovered first. But ACE indicates an enzyme. So yeah. All right. And of course, can you name those enzymes? Yeah, name those enzymes. You probably have to. Pretty soon, hexokinase is opposed by glucose 6-phosphatase. PFK1 is opposed by fructose 1,6-bisphosphatase. The neat thing is, I don't require you to memorize that whole big chart, but if you have it memorized, you can guess the enzyme name a lot of times. Fructose 1,6-bisphosphate,
Yeah.
fructose 1,6-bisphosphatase
That's what I
becomes
was
the
thinking,
enzyme.
like, if we're gonna have like a chart, because I was
Yeah.
like, I ain't gonna lie, I was just like, ACE.
Yep. Now, the weird thing is, this is fructose 1,6-bisphosphatase 1, which implies that there's a fructose 1,6-bisphosphatase 2, which is an enzyme that does another target of regulation. We'll get to that later, maybe briefly. Yeah. And then, of course, there are isomers of various enzymes as well, too, which is what we're going to talk about now. Oh, how's that for transition? It's almost like Dr. Ebert prepares for these classes. I don't know. When we talk about hexokinases, there are 4 particular isozymes that all have, they're expressed in all, that are expressed in different tissues and have different enzymatic, they catalyze the same reaction, but they have different rates in there. So hexokinase 1 is in all tissues, different levels in there. You might not have a whole lot in, skin or something like that, but you're going to have some pretty much everywhere. Hexokinase 4 is the one that we are most concerned with because it's our glucokinase. It's only expressed in the liver. So that hexokinase chart we saw there before, in the liver, it's got a very high Km. So it is very responsive to high glucose concentrations. So if we see the glucose concentrations increase, we're going to see hexokinase 4 activity ramp up in there. It's also not inhibited by glucose 6-phosphate. So even as it makes its product, that doesn't immediately feed back and slow it down, unlike some of these other isozymes where that will happen. So what hexokinase 4 is for, that's kind of weird to say that, hexokinase 4 is for, its purpose is to clear blood glucose at high concentration. So if I chug my can of Mountain Dew and snort a line of Pixie Sticks in the morning as breakfast, don't recommend that, at least not at my age. I got a ton of glucose in my bloodstream. So hexokinase 4 is just like, get it out of the bloodstream and into the liver. And again, we don't want high levels of glucose circulating in the bloodstream because that can lead to inflammation, and you see various effects that are associated primarily with diabetes, right? So we want to clear that out of there. Okay, the other isozymes are doing the same thing. They're going to have very similar sequences, but different kinetic properties, and we can regulate them differently. Okay, so if we look at hexokinase 1, it's pretty much got this basal level. It's kind of like, oh, look, there's a sugar. I'll phosphorylate it, put it over there. Oh, look, another one. It's kind of very lackadaisical. All right, whereas hexokinase 4 is kind of like, oh, more sugar, more sugar. Okay, we got to get this in here. We got to get the job done. It's our hyperactive go-getter that's going to get stuff done in there. All right, and we can see this in our, yes, yeah. And we can see that it is, initial velocity is a lot higher as you increase the glucose concentration. Whereas hexokinase 1, it doesn't matter if there's, 1,000 glucose molecules or 1. It's like, yeah, sure, pick one up in here. Yeah. Maybe it's like the, I don't know. I was gonna say, come up with your own analogy here. I was gonna come up with something ridiculous. Yeah. It's like the bouncer at the door of a club, because Dr. Ebert is so familiar with the club. It's like, okay, there's one person out there, I'm gonna let one person in. That's hexokinase 1.
Yeah.
There's 1,000 people out there, I'm gonna let one person in. I'm gonna let one person in. So hexokinase just slowly goes about that. Whereas, hexokinase 4 would be more like the stadium, open up more doors, get more people in or out. Something like Okay. So for our purposes, hexokinase 4 is more interesting because it's in response to glycolytic flux from maybe our diet. We bring in more glucose, or we have an absence of glucose, so we can turn it off and turn it on, or speed it up and slow it down is more accurate. It's regulated by sequestration and also by transcription. Okay, so if we have a lot of glucose, we're going to see hexokinase 4 move to the membrane to bring that get activated in there. We see our GLUT2 transporters move to the membrane to bring the glucose in. Our hexokinase 4 is sequestered in its own little, let's say vacuoles, endosome, something like that. And there's some little molecule in there, but they'll be released as the glucose comes in so they can act on it. We'll also see in the nucleus, there are various regulatory proteins in there. And if we continue with a high sugar diet, we'll see increasing transcriptional activity for hexokinase 4 so that it can handle more glucose from the diet. So you get that control over time, transcriptional control. What I'm looking for, adaptation to changes in diet. So what happens if you go on like a sugar-free diet, no carb diet? Well, you're going to stop making as much hexokinase, and the transcription level will go down. You'll have fewer molecules of hexokinase 4 in there.
You said is it fluid by transcription?
Sequestration within a vesicle. Vesicle, that's a better word.
It's only
Yeah. Yeah. Any type of endosome in there. Okay. All right. So, we either kind of hide it away so it doesn't act on anything, or we, longer term, we look at transcription effects. If we compare this, we can compare this with phosphofructokinase 1, or PFK1 in there. So again, it's our first committed step in glycolysis. ATP is a substrate for this. Okay, so it requires ATP to add that other phosphate group on there. So we're spinning energy, but ATP is also a negative effector. So if we have a whole lot of ATP, it can bind in the active site to transfer the phosphate, but it can bind in a regulatory site to inhibit it. Okay. Yes. Yeah. All right. Conversely, though, AMP or ADP are actually positive effectors that will speed up PFK1. Okay. Citrate inhibits it, and we see that will be coming from the citric acid cycle, and that usually indicates a healthy amount of energy if we've got excess citrate in there. And we also have this fructose 2,6 bisphosphate. Remember that fructose 1,6 bisphosphatase 1? Well, now we have a fructose 2,6 bisphosphatase. But this is just a regulatory molecule that affects PFK1. Okay, so we basically make a cognate molecule of the product here. And if we have a lot of fructose 2,6 bisphosphate, it's going to say, hey, make more fructose 1,6 bisphosphate. Ooh, man. Okay. If we look at ATP levels, low ATP levels, okay, we still need to spin some ATP, but we're making ATP through glycolysis, so it's going to enhance the activity there. On the other hand, if we have high levels of ATP, we're going to inhibit this so that our PFK1 activity is much lower, at least until our concentration of fructose 6 phosphate rises and drives it forward. Okay. So there's a whole lot of little tweaking. Should probably avoid that phrase. It's like I think of like tweaking like ham radio dials, but tweaking. You do some math to study. Yes. Yeah. So we're like whack-a-mole. I hit something down over here and something comes up over there. Yeah. That's a good analogy for studying biochemistry. It's all interrelated. Okay. So let's compare the regulations of PFK1 and fructose 1,6 bisphosphatase, the reverse enzyme. Okay. So if we look, for going to glycolysis, PFK1 is downregulated by ATP and citrate and upregulated by AMP and ADP. All right, awesome, cool. Okay, so we're making energy. If we have low energy, our ADP and AMP concentrations as they increase signal low energy, we're going to drive this forward to make more. On the other hand, if we have high levels of AMP, it's also going to bind to fructose bisphosphatase 1 and inhibit it. It's going to say, stop making glucose. We need glycolysis to make more energy, to make more ATP, to reduce these AMP levels in here. So you can see AMP has a dual regulatory function. ATP, none of the rest of these bind to it, but there are some others that would have, opposite effect. Well, there are other examples of antithetical enzymes where they'll have the same substrate will bind to both of them and promote one and inhibit the other. Okay, so I like this. Go glycolysis if AMP is high and ATP is low. Go gluconeogenesis if AMP is low. Okay, so we've kind of mentioned this fructose 2,6-bisphosphate, and it's important to note this is not a glycolytic intermediate. It's just a regulatory molecule, and it's made specifically to regulate glycolysis and gluconeogenesis. Okay, it activates PFK1 for glycolysis, and it inhibits fructose 1,6-bisphosphatase for gluconeogenesis. So if we are in a state where we need more glycolysis, we're going to make some fructose 2,6-bisphosphate to promote PFK1 and inhibit fructose 1,6-bisphosphatase. Okay. And here is an example of our differential regulation. If we look at concentrations of fructose 6-phosphate, so for PFK1 activity, we can see when we add fructose 2,6-bisphosphate, we see this increase in activity, and until we get really high concentrations in here, and we'll start feeding back to turn that off. We'll stop making that. We look at fructose 1,6-bisphosphate, and now we're comparing this to the FBPase 1 activity. We'll see that this will activate it. Okay, until we get to minus 2,6. Oh, we're just looking at our relative concentrations of our other one. Okay, fructose 6-phosphate. So that's going to feed forward to make fructose 1,6-bisphosphate. And here we have a lot of fructose 1,6-bisphosphate. We're going to push it back the other way. Okay, and that's why we see these kind of flipped around, because they are opposite direction enzymes. PFK activity going one way to make this, FBPase activity going the other way to consume this and make it fructose 1,6-phosphate. Fructose 6-phosphate. Fructose 6-phosphate. Taking off the 1. Yeah, sorry. This is where I should have spent more time prepping this slide. All right. If we look at our bottom axis, that's our concentration of fructose 6-phosphate. So the more fructose 6-phosphate we have, the more we want to convert that into fructose 1,6-bisphosphate. So that's going to drive our PFK1 activity. As we add fructose 2,6-bisphosphate, we see the activity of PFK1 increase. Okay, if we take away fructose 2,6-bisphosphate, we see it decrease until we get to some super high level of fructose 6-phosphate that overcomes that in lack of promoter and drives it forward anyway. Okay. For the reverse reaction, where we have fructose 1,6-bisphosphate and we're converting it to fructose 6-phosphate, if we take away fructose 2,6-bisphosphate, we see the activity of FPBase 1 just skyrocket, boom, up there. As we add fructose 2,6-bisphosphate, we slow down FBPase 1. So these are kind of opposite things are happening. As we add F26-bisphosphate in here, we increase this one, we decrease this one. Okay. As we take it away, we slow this one down, we turn this one on. It's almost like an on-off switch here, because you see there's almost no activity, and then at very low concentration of F2B6, bam, we get 100% activity. Okay. That's a better way of saying that. Yeah. Okay, so fructose 2,6-bisphosphate regulates fructose bisphosphatase 1, where do we get fructose 2,6-bisphosphate from? Same place we get fructose 1,6-bisphosphate from, fructose 6-phosphate. We just have a different enzyme, PFK2, because it adds a phosphate group to carbon 2. Convenient naming, right? At least the names make sense. PFK1, we add that to phosphate 1. PFK2, we add it to 2. All right. So it's another enzyme, different enzyme from PFK1, does nearly the same thing, just slightly different. Okay. So, what's the difference between vanilla ice cream and French vanilla ice cream? Okay. Or chocolate chip and chocolate. That's probably too close. Okay. And of course, the enzymes that are involved in this are regulated very similarly to the enzymes in glycolysis and gluconeogenesis as well too, right? So why do we have this extra layer of regulation in here? More control, more fine tuning. We can make that, if you go back and like watch videos on the cell phone from the 1990s, they're really grainy. And think, is this filmed on a potato? And now, of course, it's like, oh, I made this movie on my iPhone, So we're getting, just like our cameras are getting smaller and more sensitive. Our fine-tuned control of metabolism is important for response to changes in steady state concentrations. Okay. All right. So how do we regulate fructose 2,6-bisphosphatase levels? Okay. Well, we've got two enzymes here that are different than others in glycolysis and gluconeogenesis. They are conjoined. So we have two enzymes that are attached together. I got an active site here and an active site here. All right. So when I have high levels of fructose 2,6-bisphosphate that will stimulate glycolysis and inhibit gluconeogenesis, we have this PFK2 that becomes active and FBPase2, which is inactive. So it's really this, we could call it the same enzyme with two effects or two attached enzymes, okay? When we see insulin released, it indicates glucose. We want to promote glycolysis because we have lots of glucose in there. We're going to see this that's going to activate this phosphoprotein phosphatase, which will dephosphorylate this bifunctional enzyme and turn on the PFK2, the kinase part, and turn off the phosphatase part. On the other hand, if we are at low energy state, one of the hormones that we see for low glucose levels that kind of stimulates hunger as well too is glucagon, which is going to result in increased levels of cyclic AMP, which is going to cause ATP to be used to phosphorylate this enzyme, which means we will stop producing fructose 2,6-phosphate. We'll turn off the PFK2 and we'll turn on the fructose bisphosphatase 2 and convert that back into fructose 6-phosphate. That's a lot of talking about these two slides. So it's, and yeah, and this is detail. That's the crazy, the horrible thing about this class. It's detailed and everything is interconnected.
This is like a marriage.
Yes.
Like,
yeah.
Like you have a kid,
It's,
one of you guys are a morning person, and the other one is a night person. You got twins.
exactly. Yeah. One person works day shift, and one person works night shift, and yeah. Horrible. Alrighty. So, PFK1 regulation, you look at this and go, oh my God, that's awful compared to the hexokinase 4 regulation. Hexokinase 4, not too bad. PFK1, well, you got this, and you got the opposite thing, and then you got this that affects this, but this is also, so it's a lot. So let's look at pyruvate kinase, right? Our last one in glycolysis in here, not as bad as PFK1. Okay? I don't think. Okay, pyruvate kinase is allosterically activated by fructose 1,6-bisphosphatase. So if we have high levels of fructose 1,6-bisphosphate, that's our first product in the glycolytic pathway. It's going to kind of feed forward and turn on pyruvate kinase. So this is one of those rare feed forward things. Oh, we got flux coming. Get ready. All right. On the other hand, pyruvate kinase is inhibited by signs of abundant energy supply. So ATP will bind to it and slow it down. Acetyl-CoA or long chain fatty acids can also bind to it and slow it down. And alanine can do that. And like, why alanine? Well, that indicates we have a sufficient pool of amino acids. And keep in mind, we can, shunt our pyruvate into generating new amino acids. Okay. Pyruvate kinase is also inactivated by phosphorylation in response to signs of glucose depletion, but only in the liver. Okay, so if we start using up our glucose, we're not going to, undergo glycolysis in the liver. However, remember the brain and red blood cells only get energy from glycolysis. They're going to keep going. All right, so we have different isoforms that are organ specific. Okay. Instead, what will happen is, if we get low levels of glucose in the liver, we stop running that through glycolysis and we export that glucose to go to the brain, other places. So if we look at this, we can see phosphorylation, it's inactive, dephosphorylation, it's active. Right? Glucagon is going to stimulate phosphorylation to turn it off. And then, of course, over time, if we don't have the presence of glucose, our phosphokinase A won't be activated. We will instead see this pyruvate phosphatase that will dephosphorylate. Okay. We can also see our allosteric effectors in here. ATP, acetyl-CoA, long chain fatty acids all bind to it, inhibit it. Alanine feeds back and inhibits it. And F16BP feeds forward and activates it. So this is just kind of a summary of those previous two slides. Yeah. You guys like this? You guys afraid of this?
I think it's just going to be a lot of drawing.
Yeah. And that's the weird thing about having slides. I try and put a variety of slides up here so that there's a method, there's something you see that you like, and then you can mimic that on your own notes. All right. If we look at pyruvate, so we go all the way through glycolysis, we've made pyruvate. We briefly mentioned fates of pyruvate. We can use it, we can go backwards through gluconeogenesis and make new glucose, okay? And we can store energy as glycogen. We can generate NADPHs through the pentose phosphate pathway. Pyruvate can be used to make acetyl-CoA, which we can store, use in production of fatty acids and store as body fat. And we can also make ATP through the citric acid cycle. Guess what's coming up after the first exam? And of course, what's that? Yes, yeah. But that'll be the next exam material. I don't want to throw too much at you all at once. Every chapter is too much, but I have these grouped in like 4 or 5 chapters for every exam. It's a lot. Acetyl-CoA will also stimulate glucose synthesis through gluconeogenesis. Yeah, by activating pyruvate carboxylase. All right. Okay. So alternative fates for pyruvate. Go back up, make glucose. Go make acetyl-CoA into the citric acid cycle. And we, for some reasons not on here, make amino acids. All right. Okay. So, and this is kind of my bias. I grew up as a protein biochemist. I'm interested in enzymes. So I look at how enzymes bind things and regulate and stuff like that. That's only part of the regulation of these enzymes. We also have transcriptional control. So usually our protein interaction, these effectors, phosphorylation, dephosphorylation, whatever, those are faster, a more rapid response. Transcriptional control tends to be a bit slower and happens over time. So if you change your diet, you're slowly going to change what genes are being activated and turn on some, turn off others in here. So some of these can be regulated by insulin. So, if you're constantly sipping on a sugary drink, you may, constantly release insulin, and you're going to see increased expression of hexokinase 2 and hexokinase 4. We didn't really talk about hexokinase 2. Also PFK1, PFK2, pyruvate kinase, all those things in a glycolytic pathway because that indicates that you have a constantly high circulating level of blood sugar. So you want these activated to kind of clear that out. Conversely, you'll see decreased expression of Pepsi K, because if you have so much glucose in the bloodstream, you can constantly run glycolysis, and you don't need to run gluconeogenesis to make more glucose because there's plenty of it there. So insulin over time is going to result in decreased expression of Pepsi K, and similarly glucose 6-phosphatase in there as well. So that's just an adaptation. Change is hard because you got to change your DNA pattern expression. Okay, we also have this cholesterol response element binding protein that activates transcription in response to glucose in here, or we just call it CREB-P, okay, or TREB. CREB-P, I don't know. Name your child CREB-P for both now. So here we have our glucose comes into the glucose transporter, gets phosphorylated, blah, 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 goes down here like, oh, we make xylulose 5-phosphate. So we've kind of shunted that off. It's not going into glycolysis. This is another pathway in here. But what this will do is this will activate this phosphatase 2A, what was it? Something phosphatase 2A, which will dephosphorylate this CREB-P regulatory protein. As it gets dephosphorylated, and notice it takes off one phosphate but not the other. One phosphate sends it to the nucleus, where we have another isozyme of PP2A, which will take off the other phosphate, which will then, I love this, is it mix or MLX, but interacts with another, I think it might be MLX protein in here, but CREB will interact with that. Together, they'll bind to this response element in the DNA, which will affect the transcription levels of proteins that are involved in fatty acid synthesis or glycolysis. It'll basically turn both of those on, because if you have lots of glucose, you can store energy, fatty acid synthesis, and of course you want to run it through the glycolytic pathway, so glycolysis in here. So this is our detailed step-by-step how we actually do the transcription. And notice our transcription factor is kind of kept sequestered cytosolically until it's acted on, where it moves into the nucleus, where it then interacts with a binding partner, and then those binding partners together allow it to bind to a specific response element, which then affects the gene expression in there. Whew. It's a lot. Am I going to ask you to know this specifically on the exam? Not on the in-class portion. If there's a take-home part, I may ask for some big details on this and stuff, or like a similar, you see something similar to this, because this type of interaction is very common, where you'll see things are sequestered someplace, then they get phosphorylated or dephosphorylated, move someplace else, interact with something, or something comes apart. You see a conformational change, new binding partners, which allow it to then bind with something else that does something else. So, yeah. I'm gonna post a music video of a song. It's not actually not that old. It's probably like 10 years old, but it's called An Honest Mistake. It's probably not typical music here, but the video is awesome. Are you familiar with Rube Goldberg machines? Big contraptions that do a whole bunch of steps to do nothing. And then like during the early stage of the pandemic, there were a lot of viral videos. Like there's one kid who built this thing to get a basketball to go in the hoop, and it had like all these steps in there. Yeah. So it's something like that. But this video for the song basically takes that idea, but then there's a twist at the end, which is fun, which is great for studying biochemistry. It's like, yeah, it's so bitter and cynical. You got to be cynical to study biochemistry. All right. We can look at some other examples in here. I like this FOXO1 activates transcription response to insulin. So insulin will bind to it. We'll see this phosphorylation interaction here. Bonus points. Any idea what type of protein of hormone receptor this is? Maybe. Yeah. I think it's like the second or third one that we studied in chapter 11 or 12. I know it was the last section of Biochem 1. Well, so this, the integrin receptor is very similar, but I think the class is tyrosine kinase receptors.
Oh.
Yeah, so,
When I think it's certain, I always think of figures.
yeah. And the only reason I say tyrosine kinase receptors, these little lines in here, oh, that typically indicates phosphorylation. Boom, we're glowing, like turning on the lights. Yeah, so we get this autophosphorylation. Ultimately, we activate protein kinase B, which will phosphorylate FOXO1, and then it gets ubiquinated from that. But when FOXO1 is dephosphorylated by this phosphatase protein, it can then go to the nucleus and turn on transcription for Pepsi K and glucose 6-kind. So insulin says, hey, we don't need your services anymore. We're going to lay you off. We're going to lay you off. We're going to destroy you, basically. But when we don't have insulin, typically we have low glucose concentrations. So that's when we're going to need gluconeogenesis to be active. So FOXO1 transcriptionally activates the production of Pepsi K and glucose 6 phosphatase. Good times, right? All right. This is what I call a bullshit slide. This is like, in case you thought this was easy, here are, many different transcription factors that can bind with binding partners and come apart and all sorts of stuff in here. This is just, other stuff. So FOXO1 binds to place and turns on, but you've got, I think we mentioned the PPARs somewhere at the end of Biochem 1, and our SRE binding proteins that can bind to sterile response elements for like cholesterol metabolism. There's just all sorts of stuff in here. Yeah, exactly. It's like, you better study. All right, scared straight. You'll end up in biochem jail if you don't study all this. Yeah. So, and of course, because I'm a bitter, cynical person, I don't get rid of this slide in here. I'm scared, buddy. All right. But of course, I leave. It's also hard for me to throw stuff away. I'm almost like a pack rat. But it doesn't help that I have students who have come back from either like pre-med stuff or from the first year of med school or first year of farm school and say, yeah, I saw that stuff you were talking about. It's like, yeah, was it the there's a pre-med one that they run at Chapel Hill. I forget what it's called now, but one student was like, I was the only person in the entire class who knew what they were talking about for this lecture because of some silly slide I left in here. Yeah, one of the summer prep. I just forget the name. I'm blanking on the name of it right now. Of course, I had another student who was like, yeah, I was sitting there in that class and I realized I had seen this before and I wish I'd studied harder at biochem. Yeah, so yeah. I'm like, yeah. All right. So that's our transcriptional regulation in there. Let's kind of talk about something a little bit different in here. If we have glucose, it can go into the glycolytic pathway, but we can also save some for later. And it's funny to see like my kids like candy, but they don't scarf it all down at once and they will save it for later and they'll hide it from each other later. Hopefully I'll find it. Yeah. So we can save some for later as glycogen. And we've talked about glycogen in Biochem 1. It's a branched polymer of alpha-1 port-linked glucose molecules that have these alpha-1 6 linkages every 12 to 14 units. We see glycogen storage primarily in the liver and the muscle. Glycogen gets degraded. You can basically cut off a glucose as you need it. It's like a hummingbird feeder. It'll just cut some off of here. And of course, all these little branch points allow thousands and thousands of glucose molecules to be cleaved off fairly quickly. Okay. So our glucose residues are removed from glycogen by glycogen phosphorylase. All right. So we essentially cleave one off of here. We phosphorylate our glucose that we cleave off. So we now make glucose 1 phosphate, which I bet we can convert into glucose 6 phosphate. So in there, all right. So glycogen phosphorylase works on the non-reducing ends until it reaches a branch, 4 residues away from the branch point. And then we have a debranching enzyme, which is going to take that branch point and put it on the end here. And then we can cleave it off. Well, 3 of them on the end here and cleave off the branched one. And then we continue on again and just repeat this over and over again. So glycogen phosphorylase and a debranching enzyme when we get down to a branch point or near a branch point. And of course, we have to isomerize our glucose 1-phosphate into glucose 6-phosphate. And this happens through a mutase. I like the mechanism in here. We have a phosphorylated serine. So usually our serines, that hydroxyl group on the residue, is a good target for phosphorylation. Phosphorylation turns it on. We transfer that phosphate group to the carbon number 1 of the glucose 6-phosphate. And then we refresh the enzyme by transferring the phosphate from carbon 6 back to that serine. Okay, we're just gonna twist it, a little bit, put that in there. Okay. So we've talked mainly about glucose in the bloodstream, gets into the liver, we phosphorylate it so it can't get out. All right. Well, if we go through gluconeogenesis and we make our glucose, we got to get it out. We make our glucose 6-phosphate, we got to dephosphorylate it so we can get it in the bloodstream and get it to the brain. Brain need. Gonna hit my sugar high here and go, yeah. Okay. So our glucose 6-phosphatase is sequestered in the endoplasmic reticulum, and it allows the use of concentration gradients for glucose and glucose 6-phosphate to control the flux of glucose out of the liver. Okay. So our glucose 2 transporter transports either way, just wherever the concentration is there. So here's our cytosol. Here is our ER lumen. We can bring in our glucose 6-phosphate, have our glucose 6-phosphatase make glucose, which we then transport right back out of the ER aluminum close to the GLUT2 transporter, sends it into the bloodstream. Okay. And our pyrophosphate that comes off here as well too gets transported out here as well. So our glucose transporter and our phosphate transporter are kind of tied together. But, be an example of Symport. Okay. All right. So if that's cleaving our glycogen, how do we actually make the glycogen? Well, it's going to be a multi-step process. Almost everything is a multi-step process. It's like on the assembly line. Henry Ford would be so proud of the human body. So the synthesis of glycogen requires more enzymes than just our phosphorylase and our debranching enzyme. So blood glucose has to be phosphorylated, hexokinase. It has to be labeled with UDP or uridylated, and then it has to be actually enzymatically added to the glycogen. So we got several steps. We got several places where we can regulate it. Oh no, right? So glycogen is synthesized by glycogen synthase. At least the name's pretty easy. And again, another A's, right? Another enzyme in there. We're going to take uridylated phosphoglucose in here. So basically have UDP UDP attached to our carbon 1. Okay, we're going to cleave off the UDP and we're going to add it to our non-reducing end to elongate this chain. Okay. All right. UDP glucose, and this is one of the few times we'll talk about UDP, UTP, and all that stuff in there too. It's substrate for glycogen synthasase. Here we have our uridine. What we get here is a triphosphate sugar phosphate. Yeah, our sugar phosphate is going to attack the phosphate group of our UTP, transfer one of those phosphates there, and the pyrophosphate that gets cleaved off is going to be transferred. It's going to be cleaved off as pyrophosphate, which can be converted into inorganic phosphate through inorganic phosphatase. But we now have 2 phosphate groups that attach our sugar to our nucleotide base. Okay. All right. So that's why we'll often see our sugars will have an NDP, nucleotide diphosphate sugar. We're cleaving off 2 phosphates, but the phosphate group of a phosphorylated sugar attacks the nucleotide triphosphate. Right. And again, look at this. It's another serine. Similar step. All right. All right. And it's, of course, all this is reversible. Okay. So that's good. We can add this along here and make a big long line. If we were potatoes, we could stop here because that's basically amylopectin. Sorry, amylose, that's unbranched. but we make branch points every 12 to 14 residues in glycogen. So we need a branching enzyme. All right. So that can be regulated as well too. So our branching enzyme, dang it, come back here. Our branching enzyme will cleave off a section and attach it, as an alpha-1,6 linkage in here. Okay. So we basically just kind of make a little cut and put this together to make a branch point. But just like we have a debranching enzyme, we have a branching enzyme. Suppose we use up all our glycogen. How do we make it from scratch? It's kind of like your grandmother's pie crust recipe. You got to make it from scratch. But we need glycogenin. Genesis start glycogen genesis. So glycogenin is going to be our initial compound, and then we'll see our we'll extend. Again, we're using UDP glucose in here. We repeat this several times to get us a nice little starter. So here's our glycogenin, our first little branch point. We get 6 glucoses in there, and then we can start making branch points. We can cleave off 4 and put them in there, and we start another branch, and so on and so on. So we make this big granule of a glycogen particle. And you can see there are lots and lots of exposed ends. So there's lots of places where you can cleave off of glucose. And if we just look at this one, there's probably like 2 to 3 dozen glucoses you could have immediately from enzymatic activity. So you can get a lot of sugar real fast. Okay. Maybe it helps to be biochemistry and be like a little kid with a sweet tooth. It's like, I need a lot of sugar. Okay. All right. So how do we integrate glycogen synthesis and degradation? Because again, all of our enzymes die over time, and you saw the liver tends to have the fastest. Pretty much within a day, everything turns over in there. So glycogen synthesis and degradation is fairly quick. We look at the regulation that occurs at irreversible points. Okay, so we have lots of little steps in there that are reversible. We're not going to sweat that. You'll see in this class a whole bunch of little arrows. Yeah, there's many steps, many steps. We're just going to focus on the regulated ones. Okay, so our UDP glucose pyrophosphorylase, our glycogen synthase, our glycogen phosphorylase. I want to take a peek here real quick. Make sure I'm not going too far. Oh, okay, good. I think we're where we need to be. How do we control glycogen breakdown? Okay, so this is not immediate, like I eat a whole bunch of sugar and I need to turn on glycolysis, or oh yeah. This is a little slightly longer pathway here. Well, except for epinephrine, but a glucagon epinephrine signaling pathway will start the phosphorylation cascade in there. Adrenaline functions like epinephrine in there. And it will activate glycogen phosphorylase, so we'll start cleaving off those ends. So glucagon is like, oh, I'm hungry. I'm kind of in a low energy state, so we'll cleave it off there. Epinephrine is, bear, fight or flight response. So it's going to activate it a lot faster in there. Glycogen phosphorylase cleaves off those residues and gives us our glucose 1-phosphate, like we saw in there. And I think most of you have seen this picture before. Epinephrine, we see this cyclic AMP as a second messenger, which will activate protein kinase alpha, which will activate phosphorylase B kinase, which will activate another kinase, and so on and so forth. And all of a sudden, we've got 10,000 molecules of glucose, so that you can fight off a bear with your bare hands. Notice the pause. Okay. Yeah, that's true. That's true. Should I think? Yeah. Side note. All right. You ever been to Great Wolf Lodge? Yeah, it is expensive. I remember when my son was born, we looked at it, and like every couple of years I'd look at it and go, that's expensive. That's expensive. Apparently my wife makes a lot more money than me, so we took the kids to Great Wolf Lodge last year for a couple of days. It's weird. It's yeah. I mean, it's fun. It's got a nice water park, and that's what the kids were there for and all that. But then they try to make it very family entertainment and all. But it just struck me as really weird because everybody gets wolf ears when you get there, and you can get like the wolf paws and the wolf tail and the wolf. And I'm like, is this where furries are made? So yeah, it's unusual, and a lot of the I don't know. I just found it outside my normal range of entertainment. So it wasn't bad. They did what they did well, but I was just, this is not me. So Anyway, back to this. Again, our glycogen synthase controlled by glucose uptake and phosphorylation. Actually, let me check the time. Yeah, we might. Yeah, if I'd gotten here, gotten the Wi-Fi to work. Anyway, glucose comes in through the GLUT4 transporter in this case. We go through here and we can see our insulin activating our tyrosine kinase receptor. So there's a lot of crosstalk in here as well. So, if we have lots of glucose, we can go to glycolysis. We can also store that as glycogen. Glycogen synthase is controlled by phosphorylation. So as we phosphorylate it, we inactivate it. And as we dephosphorylate it, we activate it. Okay. Don't want to say more about that. So the glucagon epinephrine is going to inhibit the activation of glycogen synthase. Insulin, glucose 6-phosphate, and glucose are all going to promote the activation of glycogen synthase. So, and I should say, notice the way I said that. They don't directly activate glycogen synthase. They promote the phosphatase that removes the phosphates that then activates it. So there's they bind to the phosphatases in there. Okay, so to summarize all the stuff we've been talking about, if we have high blood glucose, we're going to see increased insulin in response to that, which means we're going to see increased insulin sensitive protein kinases, protein kinase B, protein phosphatase 1. We're going to see synthesis of hexokinase, phosphofructokinase 1, pyruvate kinase, all these things that lead toward increasing glycolysis, increasing glycogen synthesis, and decreasing glycogen breakdown. So our protein phosphatase 1 will dephosphorylate phosphorylase kinase, which will slow down glycogen phosphorylase and inhibit glycogen breakdown. Okay, so high blood glucose, insulin, all this kind of leads to these factors. Conversely, low blood glucose is going to see the release of glucagon, which is going to bind to its receptor and make cyclic AMP, which will increase levels of protein kinase alpha, alpha beta, wait a minute there, yeah. And we'll see increased phosphorylase kinase activity and changes in pyruvate kinase, decrease in pyruvate kinase, decrease in phosphofructokinase 2, increase in FBPase 2, so that we will inhibit glycolysis, we will inhibit glycogen synthesis, and will promote glycogen breakdown. Okay. So depending on our fed state or starved state, we avoid a futile cycle this way. That's what we're doing here. All right. We talk about the liver a lot, and that's important. What we see, though, is that different organs have different responses to the same stuff. So epinephrine and glucagon in the liver, we see all this, breakdown, glycogenolysis, reduced glycolysis, increased gluconeogenesis. And here, if we have lots of pyruvate feeding back in this way, pyruvate is going to stimulate our gluconeogenesis, and it's going to inhibit glycolysis. Except in the muscle, pyruvate is going to stimulate glycolysis because it indicates we're at a low energy state and we need more of that. Okay, glycogenolysis with our epinephrine is going to be increased in here as well too, so that we can make our muscle glycogen. Now, muscle glycogen is weird. You got glycogen in your muscles. You can use that for energy. You almost never use that for energy unless you are nearly exhausted or greatly starving. Even when your glycogen stores in your liver are massively depleted, you are still cycling back to make more glycogen in the muscles.
Is that why they say, like, when you just sit there, like you say, it's starving, you'll lose, like, you have to think, well, sometimes they say it's first to go.
Yes. So it depends. It kind of depends on what you need there too, because muscle breakdown will occur. When you're depleted of energy, you're going to start breaking down those amino acids in the muscle too for energy. But usually right before that, you've broken down the glycogen that's in there. The glycogen is kind of like your last ditch stand. Yeah. So if you're exerting, you're using a lot of energy, you might be pulling glucose from the glycogen in the liver, but your muscles are like, nah, it's not that bad yet. Nah, we're still fine. Nah, we're good. When you get into when you get into really dire straits is when you start using muscle glycogen. And you may actually use some of that muscle glycogen in your exertion, but you're also spending some energy to immediately replenish it. It cycles back through there. It's weird because when we get to the fatty acid metabolism, you're going to see that something like 70% of the triacylglycerides that are cleaved are immediately um, reformed within the adipocyte. So there's a flux in there that kind of goes back and forth. Even in like a energy deprived state, you're constantly cycling back in because it's apparently incredibly important for the survival of the organism to store some energy in these places. Yeah, and it's just weird in there. All right, we won't talk about this today, but we try and make this fun. I will make this fun for Tuesday. We'll do a little bit of review, but we'll also talk about some of these diseases in here. Some of these you may have heard of. Pompei's disease is not terribly uncommon. Cori disease is out there, but some of these like Harr's or Tarui, you almost never see. They're just various in there. I do like to pull case studies from that national database of rare diseases. So we will look at some of these glycogen storage diseases. You can see we have, a dozen easily of these. And typically you'll see things like enlarged liver, because, you're trying to produce glucose constantly in there. You may see low blood glucose in type 0 where you just can't make any glycogen synthase. So you make a lot of high ketone bodies and type 0 bad. Kidney failure for von Gierke's. What's a Pompei's disease? Infantile form, death by age 2. Also really bad. If you can't store, depending on, which particular enzyme is being affected, this is rapidly lethal. Or, you'll see a lot of things. I'm trying to figure out where's the one. I think it's in here. Myopathy, Cori disease. You tend to get people who just don't have very much muscle mass at all. And you'll see these really skinny look. The comparison would be like if you've seen pictures of survivors of the concentration camps in World War II that were horribly underfed. You see people who have muscle tones similar to that, just because they can't, there's no debranching enzyme in there. So, all right. And that's it for this chapter, but we'll do some fun stuff. Yeah, fun stuff. We'll talk about terrible diseases. That'll be our fun stuff that we'll do on there. But the biggest thing out of this chapter is we're really kind of tying together our introduction in here of metabolic flux. Things are constantly changing. We eat food. We go a period without eating food, like when we sleep at night. How does the body change in response to the levels of glucose, or as we'll see later on, fatty acids or proteins, depending on what we eat? So we'll do a bit of a review and some fun stuff with those diseases in there. I don't know that I'll plan for the test for next Thursday. I have to look at what I want to do. I think I usually like to give exams on Tuesdays because there will be a take-home portion. Oh, no. Yeah. Oh, you got, yeah. Let me know about what other exams you have as well too. Like when is this coming? Yeah. Okay, this coming Tuesday? Yeah. This would be the following Tuesday. So yeah, not this Tuesday. Yeah. We got to do a little review first.